This is part 2 of Chapter 45's lecture. And so we left off talking about water-soluble versus lipid-soluble hormones and their pathway into getting a response from a cell. So this diagram shows you in an overview how a water-soluble hormone is going to bind to a receptor on the membrane of the target cell. So it's coming from an endocrine tissue of some sort, endocrine cell, which is going to release the hormone into the extracellular fluid. It will make its way into the circulatory system, and it will then be distributed to a lot of different cells, but those cells that do have the correct receptor protein on their surface will allow that hormone to bind and start a signal to get a response. And that's in contrast to the lipid-soluble, which again is going to be diffused this time through the membrane of the endocrine cell into the extracellular fluid. It will go into the bloodstream, similar to the other hormones. But this time when it gets to its target cell, it's actually going to be able to diffuse into the cell and then act in some way with a receptor within the cell, either in the cytoplasm or the nucleus. Most often it is the nucleus. Okay, so this is an example of a water-soluble response. And so epinephrine is, again, one of the protein hormones. It is actually an amine, and it has multiple effects, but it has to do its action by way of binding to a receptor protein on the membrane of its target cells. And even though there are a number of different types of target cells, they are going to have some sort of receptor. So depending on where or what type of cell it binds to, so for example, we'll say this is a liver cell. So epinephrine will bind to the receptor of this liver cell. It will begin a G protein coupled relay message within the cell, signal transduction pathway. So you can see here where we're using secondary messengers and activating a kinase in order to get what we need, in this case, either the inhibition or the promotion of glycogen breakdown. The benefits of using this cascading pathway is, or what we, again, going back to our cellular communication unit, a secondary messenger system, is that we get amplification of the message, and then that way we're going to get a larger response and a quicker response. So the benefits of these protein hormones is that they do use the signal transduction pathways that amplify the message through the cascading response. So this is an animation again from the textbook of the action of a water-soluble hormone.
Hormones act as chemical signals, but how do they trigger changes in particular target cells? Non-steroid hormones, hormones derived from amino acids, oxytocin and epinephrine for example, work via signal transduction pathways to activate or inhibit proteins within cells. These hormones can trigger an immediate response in target cells by turning cell components on or off. A non-steroid hormone binds to a receptor protein in the plasma membrane. The receptor activates a signal transduction pathway in the cell. A series of relay molecules transmits the signal to a protein that carries out the cell's response. In this case, an enzyme is activated. A different non-steroid hormone might trigger a different response, perhaps turning on specific genes that result in the synthesis of a particular protein.
Okay, so just again a quick overview of a non-steroid or a protein hormone. This is in contrast to the lipid-soluble hormones. So the response to a lipid-soluble hormone is usually going to be a change in gene expression. Primarily, it's going to either turn on or turn off the transcription and translation of genes. So when a steroid hormone binds to its cytosolic receptor, which means a receptor located within the cell, the hormone receptor complex forms and moves into the nucleus. And within the nucleus, the receptor part of the complex will act as actually a transcription regulator or transcription factor for a specific target gene. So we're going to, move on here to the next slide. This is a graphic showing that, for example, estradiol, which is related to estrogen, is a hormone that can pass as a steroid through the plasma membrane. And then within the cytoplasm, there's a hormone receptor complex right here. And so once that occurs, the hormone receptor complex will move into the nucleus where it will target the DNA and make the DNA, then make an mRNA for this particular protein here. So you can see here, then this mRNA will travel back out to the cytoplasm where we'll have the translation and production of our new protein with the help of the ribosomes. So this is an animation now contrasting the lipid-soluble hormones.
Steroid hormones such as testosterone or an estrogen acts by entering a target cell and turning genes on or off. Steroids usually act rather slowly because it takes time for the gene products to build up or become depleted. This steroid hormone passes through the plasma membrane and binds to a receptor protein in the cytoplasm. The hormone receptor complex enters the nucleus and attaches to specific regulatory sites on the target cell's DNA. There it acts as a transcription factor, a gene activator. A messenger RNA molecule is transcribed, which is then translated into a protein, an enzyme, for example. This new protein alters the function of the target cell.
So another factor to consider when we're talking about the endocrine system is that one specific hormone can have a number of different effects. And we mentioned this a little ways back, but I just want to reemphasize the fact that the same hormone can have a different effect on the target cells, either because the target cells have different receptors for that hormone or because they have a different signal transduction pathway. So the example that we have here is of the hormone epinephrine. And epinephrine can cause an increased blood flow, for example, to a major skeletal muscle, but it can have actually the opposite. So in this case, it's dilating the blood vessels. In this case, if it moves over to this, it can actually constrict the flow of blood if it's in an intestine. So depending on where the receptor has been located, in this case we have two different receptors. There's a beta receptor or an alpha receptor on the muscle cell, and it's going to cause either that muscle cell to relax or to constrict or contract. So this is a case of different receptors. Other times we're going to have the same receptor but have different proteins within the cell. So in this case we have a liver cell versus a muscle cell. And in the case of the liver cell, we end up with a level of glucose being affected by epinephrine. So blood glucose levels can actually increase when epinephrine is attached to a beta receptor. So the glycogen deposits are released and glucose levels rise in the blood. Here, again, the beta receptor here on the muscle cell is going to have a totally different effect. But the same hormone is having all these different reactions or responses. This is a diagram, and I'm sure there are many that you can take a look at. And you don't have to memorize every single gland, but you really should know some really basics and be familiar with some basics here. So do take a look at this and study it, at least in a cursory way. But one of the things I want to point out is that all of these glands here are called endocrine glands. And endocrine gland, like a thyroid or a parathyroid gland, is a gland that's going to release the hormone within the body. So it's going to circulate within the body. In contrast, we have exocrine glands, which are still going to secrete a hormone, but in this case, the substance that's being secreted is its actual target is outside of the body or onto the body surface. So an example of an exocrine gland is a salivary gland, which is going to excrete chemicals out of the body. So exocrine versus endocrine. Make sure you understand the difference between the two. Now we've been talking about one of the main functions of hormones is to maintain homeostasis. So we can't really look at that or address that without thinking in terms of a feedback model. And so most hormones are going to work with a negative feedback model. And a simple model is shown in this diagram here, where we have a specific body condition, which we would call the set point for the body, which is the place where homeostasis is maintained. Either that set point the body can rise above that set point, or it can fall below that set point. And in either case, that serves as negative feedback because we've moved away from where the body is supposed to be. And so in response, we have a gland which secretes a hormone, which then lowers whatever this particular condition is and returns it back to its homeostatic point, its place where it's at its set point. In contrast, we can have, if the condition falls below what it should be, we could have either the same gland or a different gland release a different hormone, which will raise that body's condition back to the set point. So we have a coordination of different hormones acting in a way in a negative feedback loop in order to maintain homeostasis. So let's look at a specific example here. So if we have a simple endocrine pathway, we have here where the set point should be. And when we get, for example, this example would be with secretin. So, for example, when acids from the stomach are released into the duodenum, that lowers the pH in the duodenum. And then the S cells of the duodenum send a message, which is the stimulus, to an endocrine cell, which releases the hormone secretin. And that hormone then is going to travel through the bloodstream and circulate around the body until it reaches the target cell, which is the pancreatic cell, so a cell within the pancreas. And what that tells the pancreas to do, this hormone stimulates the pancreas to release bicarbonate. And that bicarbonate, in turn, will raise the pH within the duodenum. So in that way, our response is returning our body system back to the pH where it needs to be, so getting back to its set point, and the negative feedback that goes back here turns